When we look at a synapse, we're going to assume that what we're talking about is the synaptic terminal of one neuron synapsing onto the dendrite of a second neuron. All right, and what happens at this junction? So we know already that what's sitting at the postsynaptic cell are a bunch of chemically or ligand gated ion channels. And when neurotransmitter is released from the presynaptic terminal and binds to these ligand gated channels, you're going to open up that channel and allow an ion to go through. And the ion that goes through is going to create what we call a synaptic potential. All right, now synaptic potentials are graded, which means that they can start at resting -70, but you let some ions in and it goes up to, let's say, -60, for example. But if you open up more channels, it goes to -57, and so on. So they're graded, they go in steps. Now, synaptic potentials not only are graded, but they can either depolarize or hyperpolarize. And hopefully by now you remember that when you open up a sodium channel, chemically gated sodium channel, and sodium rushes in, that will always result in a depolarization. In other words, the postsynaptic membrane is going to become more positive. So it's going to go from -70 to -50 to -40 and so on. We call this depolarization by opening up chemically gated sodium channels at the synapse excitatory postsynaptic potential, or EPSP. Again, we open up sodium channels to depolarize the membrane. They go in a stepwise fashion until you get to threshold potential, and then you send another action potential down the next neuron. All right, but if we happen to open up potassium channels in this postsynaptic membrane, we'll do those in green here, then potassium is always high inside, so potassium is going to rush out when we open up those channels. And if potassium rushes out, the inside of the cell is going to get even more negative. So it's going to go from -70 to -80 to -90. And when it does that, it is said to hyperpolarize. And when the next cell in a synapse, the postsynaptic cell in a synapse, hyperpolarizes, that is an inhibitory postsynaptic potential, or IPSP. And it's a way to inhibit the next neuron so that it doesn't fire. And many of neurons in our nervous system are set up that way so that one neuron opens up channels that will inhibit the next neuron from firing through inhibitory postsynaptic potentials. And again, the reason why it's inhibited is if you're making the cell membrane more negative, it's going to be that much harder to activate it and send an action potential. So a way to look at that is if you have a neuron coming down and it synapses on the next neuron, you also might have another neuron coming in here and synapsing on that same neuron. Let's change the color to red. This neuron right here will be the activating neuron for this next neuron. This will be the, we'll call it activating, this will be the inhibiting neuron for this neuron. So going down this pathway, you cause activation, but going down this pathway, you cause inhibition. And this is a way to finally regulate how our nervous system sends signals throughout. So every time we add a synapse into a circuit, we introduce a delay. And there are 5 time periods in synaptic transmission that result in a delay of the signal. One is you have to have exocytosis of the neurotransmitter into the synaptic cleft. That takes a little bit of time to activate those vesicles to cause them to release the chemicals into the synapse. those neurotransmitters have to diffuse to the postsynaptic membrane and then bind to the receptor. Again, it takes more time than an electrical transmission. The receptor has to change conformation to open and allow the ions, whether they be sodium or potassium, to go through. We're talking about ESP, so EPSP, so that would be sodium. And then you have to accumulate enough sodium in the postsynaptic neuron to reach threshold potential. So all of that is going to result in about a 0.5 millisecond delay. So to transmit a signal from one neuron to the next via chemical transduction takes about 0.5 milliseconds. And the reason we have mechanisms like this is so that we can exert some level of control over how neurons are firing. There are many different types of neurotransmitters. In fact, there are more than 40 different types that you can find just in the human nervous system. And some of them are named here. One of the more common ones that you will probably remember is acetylcholine, and of course those are involved in muscle contractions, but you also find them in the central nervous system, involved in learning and memory. And if you remember, these bind to cholinergic. Acetylcholine binds to cholinergic receptors, and they can be of the fast type, which is nicotinic, or the slow type, which are muscarinic. And again, the reason why we have this nicotinic or muscarinic names is because there are other chemicals, such as nicotine or muscarine, that bind to these receptors as well and activate them in the absence of acetylcholine. We have monoamine neurotransmitters. These would be these are derived from amino acids, mostly tyrosine. You have your epinephrine, norepinephrine, dopamine, and serotonin. And notice that the slide shows whether these neurotransmitters are excitatory, meaning they open up sodium channels, or inhibitory, meaning they open up potassium channels. Okay, and serotonin happens to be inhibitory. Then amino acids can also act directly as neurotransmitters. So we have glycine, that's an amino acid, a single amino acid that goes into building proteins, but it can also serve as an inhibitory neurotransmitter when it binds to glycine receptors. You have glutamic acid, or glutamate, binds to glutamate receptors, different types of glutamate receptors, NMDA, AMPA, kainate, group 1, found in different regions of the nervous system and perform slightly different functions. And then GABA, which is gamma aminobutyric acid, which will bind to GABA receptors. And if you notice, there are different types of GABA receptors, GABA A, GABA B, and all GABA are inhibitory. Neuropeptides can act in the central nervous system directly as neurotransmitters, where they will produce slow responses, because what they do, instead of opening up ion channels, they activate G protein coupled receptors, which is a much slower response, or they act as neuromodulators, which alter a neuron's response to neurotransmitters. So in other words, these will affect how the ligand-gated channels will open or close. Examples of these are enkephalins, which bind to opiate receptors. And of course, these are involved in pain. Beta endorphins also bind to opiate receptors, or substance P. And notice that all of these are tend to be involved in pain. So it seems like neuropeptides are involved in the pain response. So you'll see them modulating pain in the central nervous system. And then there are neurotoxins that can modulate a neuron's response to neurotransmitters as well. So an important thing to remember is that the central nervous system, which you see in green right here, is involved in integration. So it takes many different sensory inputs puts them together in a comprehensive and understandable way, and then spits out some sort of output through the through the efferent or motor nervous system. All right, so in order to do this integration or the processing of that information, these multipolar neurons within the CNS have to be organized into circuits. All right, and by definition, a circuit is a group of neurons that work together by making synaptic connections with each other. And they do that in order to perform some common functions. And then neural circuits can be organized into pools. So a way to look at this is you have one neural circuit, which is a bunch of interneurons that are interacting with each other that go to perform one function, and another, we'll do that in red, neural circuit pool that goes to perform another function, but there may be some overlap between those two pools of circuits that go to a completely different function or something that enhances one of these other two functions. So it can get pretty complicated, and to go into the complications of that is beyond the scope of this particular lecture, so we're not going to go into any more specific detail about that right now, except for what's coming in the next couple slides. So two general types of neural circuits that you'll find are converging circuits and diverging circuits. Converging circuits is where you have a bunch of neurons coming from different sources that all converge onto one specific neuron to give some sort of output. And again, if you think about it, this is a way of integrating information. So maybe you have some input coming from olfaction, smelling. Maybe you have some coming from taste. You have some coming from texture or the different touch receptors that may be in your mouth, and another one coming from memory circuits. And they all converge onto this one neuron to give us some sort of output, and maybe that output is a feeling of pleasure from whatever you're eating, for example. Diverging circuits, on the other hand, is where you have one neuron, and remember, axons don't necessarily have to be one single unit. Axons can split into many different directions and synapse on many different other cells. So you have one neuron that's splitting and synapsing onto two neurons. These two neurons are then each synapsing onto three neurons, so now you have a total of 6 neurons being activated. And pools like this are typically used to amplify responses. And one example of this would be a motor unit that is synapsing on multiple muscle fibers in order to stimulate those muscle fibers to contract. So two other ways that nerve impulses are processed are these concepts of facilitation and inhibition. Now, facilitation is a concept where if we have nerve fiber 1 here, it has enough synapses on postsynaptic fiber A that it will trigger an action potential. But if you notice, its synaptic connections on nerve fiber B and nerve fiber C are very small. And usually that number of synapses will not be enough to trigger an action potential in B or C. However, if another neuron, let's say neuron 2 here, has projections onto nerve fiber B and nerve fiber C, and it also fires at around the same time that nerve fiber 1 fires, then it will contribute to neuron 1's facilitation of the firing of fibers B and C. So in other words, facilitation means you make it more likely that the postsynaptic neuron will fire by introducing a depolarization event that another neuron can take advantage of to start an action potential in the postsynaptic neurons. The other concept of inhibition, I've kind of already alluded to in a previous slide. We have an input fiber coming in, and it synapses onto a postsynaptic fiber, releases sodium or open sodium channels, and results in excitation. But that same fiber could also synapse onto an inhibitory neuron, so it would actually excite this inhibitory neuron, so open up sodium channels. But then this inhibitory neuron would then synapse onto a third neuron and open up maybe potassium channels on this third neuron. And if it opens up potassium channels, then this third neuron will never fire. So this pathway would be an inhibition pathway, whereas this pathway would be an excitation pathway. And this is a way that even one nerve fiber can control two different pathways, one leading to excitation and the other leading to inhibition. One example of where you'll see something like this inhibition processing is in motor reflexes, where in order to, let's take a leg here, and I am not a good artist, so please forgive my leg. In order to move a leg away from a stimulus, you will have to contract some muscles and inhibit other muscles. So in order to do that, you may have one nerve fiber that is going and activating contraction pathways and another that is going to activate inhibiting pathways. So these muscle fibers contract, but these muscle fibers do not, because if these muscle fibers contract, they would oppose the pulling away from the painful stimulus.